Alright, in this video I will explain the potato osmosis practical, which is used to investigate how water moves in and out of cells via osmosis. This practical is probably the most common required practical that appears on the exam, and essentially you just watch potatoes bulk up with water or lose water and become a raisin. First of all, what actually is osmosis? Osmosis is the movement of water molecules through a region of higher water concentration to a region of lower water concentration to a partially permeable membrane. Sure, you've heard that before. Partially permeable membrane is a membrane in the cell that prevents big molecules from entering and allows small ones, like water, to pass through. Think of it as a sieve, but on the microscopic level. This basically means water can move into and out of the cells depending on the concentration of the surrounding solution. Now for the equipment. For this practical, you will need potato, obviously, a cork bearer or any uniform cylinder-shaped thing to cut a cylinder shape from the potato, a mass scale to measure the mass of the potato stick, a ruler, salt solution ranging from 0 molar, also known as distilled water, to 1 molar, a stopwatch, or a phone, a white paper towel, and some beakers. We also have to take into account the variables. There are 3 variables we have to consider. The independent variable is what you change during the experiment. In this case, it's the concentration of the salt solution. The dependent variable is what you measure for your conclusion, such as measuring the mass of the potato before and after the experiment. And lastly, the control variable. This variable, as the name suggests, you have to control to make the experiment fair and ensure the results are actually caused by the salt concentration. And by control, I mean keeping certain conditions the same for every experiment. For example, keeping the temperature constant 20 degrees Celsius for all specimens, as a boiling potato would change the results a bit more than a frozen potato, as heat alters kinetic energy in atoms, causing the rate of reaction to change, yada yada. Or keeping the potato submerged in the solution longer than the others, which would allow more osmosis to occur, making the comparison unfair. While some variables can be quite obvious to keep constant, there can be some quite specific ones that you may not know will still affect results, such as, the type of potato used, since different potatoes have different starch contents. It can affect the rate of osmosis, so keep that in mind. Basically, if you don't control these variables, the experiment is cooked. Now, for the actual method. First, you need to cut several potato cylinders using a cork borer or knife. Make sure they're all the same size and length, because if one potato stick is way bigger than the others, it's naturally going to gain or lose more water and completely throw off the results. Next, use a scale to measure the starting mass of each potato cylinder and record the results. After that, prepare several beakers containing salt solutions of different concentrations and label them clearly so you don't accidentally mix them up halfway through. Then, place one potato cylinder into each solution, ensuring it's fully submerged. Leave them in the solutions for the same amount of time, usually around 5 minutes. Keeping the time the same is important because osmosis requires time to happen, and leaving one potato in longer than another will make the test unfair as it would absorb more water, as stated earlier. After the time is up, remove the potato cylinders and gently dry them with paper towels. This step is very important because if extra solution is left on the outside, the mass readings will be inaccurate. Finally, measure the final mass of each potato cylinder and record the results. Then compare the starting and the final masses to see how much water moved in or out of the potato cells. You can also calculate the percentage change in mass, make the results easier to compare between the different solutions. If you have seen the potato's masses increased, the potato has gained water because water moved into the cells by osmosis and is thus hypertonic. If the percentage change is negative, the potato lost water because water moved out of the cells into the surrounding solution and is hypertonic. If there is little or no change in the mass, this means the solution is isotonic to the potato cells. In other words, the water concentration inside and outside the cells is balanced, so water molecules move in both directions equally and there is no net movement overall. Overall, this practical demonstrates how differences in salt concentration cause water to move across partially permeable membranes, leading cells to either gain water and become turgid or lose water and become flaccid.